this video, we're going to cover fertilization, the process by which a sperm cell fuses with an egg cell to produce the zygote. We're going to break down the events that occur in fertilization, how the sperm is prepared, how the sperm and egg combine, and how the zygote is formed. Let's get started. So fertilization occurs when the sperm and egg cell and oocyte combine and their nuclei fuses. Because each of these cells, the sperm and egg, are haploid cells. There's only one set of chromosomes, so they only contain half of the genetic material needed to form a diploid cell, a zygote. So the zygote contains 46 chromosomes, 23 chromosomes from the maternal side and 23 from the paternal. And fertilization also triggers metabolic processes that initiates embryological development. Now, before we break down how fertilization occurs, let's first take a look at the gametes involved, their structure, and how these reproductive cells are transported. Starting with the egg cell, oocytes are non-motile, and if we refer back to oogenesis, the secondary oocyte is released from the ovary during ovulation, 24 to 36 hours after the LH surge. Secondary oocytes contain 23 two-chromatid chromosomes, but they aren't identical because of the crossing over that occurred just before the primary oocyte was arrested in meiosis 1, prophase 1. Secondary oocytes are arrested in metaphase 2 of meiosis 2 until fertilization occurs, immediately after the sperm penetrates the egg. So here's the cytoplasm of the secondary oocyte and its nucleus. Now, the egg is surrounded by two protective layers. The outer layer is called the corona radiata. It's a layer of granulosa cells. So recall from the ovulation lecture, granulosa cells support the developing egg in the ovary and secretes estrogen with the help of theca cells. And the second layer is known as the zona pellucida, which is a thick layer made up of a carbohydrate protein complex. It's a glycoprotein coat that has three glycoproteins, ZPG1 to 3, and it functions as receptors for sperm surface proteins. So the sperm head binds to the several sperm receptors on the zona pellucida. They attach to ZP3. And these layers, these envelopes, need to be penetrated by the sperm. And the sperm contains organic catalysts, enzymes, digestive enzymes that break down these envelopes, these layers, creating a pathway to reach the egg, the plasma membrane of the egg. The first sperm to penetrate the entire zona pellucida and reach the oocyte's plasma membrane will fuse with this membrane. And underneath the egg's membrane are these cortical granules. And we'll talk about these granules later because they play a crucial role. So how is the egg transported once it's released from the ovary? Let's go through this. During ovulation, the Graafian follicle ruptures and the egg is ovulated. And the ends of the fallopian tubes, the fimbriae, which are these finger-like projections, are lined with ciliated epithelium. And so the cilia start creating currents, waves that sweep the egg into the fallopian tubes as soon as the egg is released onto the surface of the ovary. And within the fallopian tube, the cilia of the tube will move the egg, the secondary oocyte, to the ampulla, and it takes about 4 days to reach the uterus. The egg remains viable for only 24 to 48 hours. It's got a best before date. And so for fertilization to occur, sperm must be released into the female reproductive tract between 5 days before and 1 to 2 days after ovulation, because the egg can only survive for about 24 to 48 hours. So that's the female gamete, that's the oocyte. Let's now talk about the sperm, the male gamete. So unlike the oocyte, sperm are motile cells, and they have three components. There's the head, which contains the haploid nucleus, okay, and this contains the genetic information, and above the nucleus is a membrane-bound vesicle, known as the acrosome, and it's a protein-filled vesicle which contains several enzymes. The next component is the midpiece, which contains mitochondria, because the sperm needs ATP, it needs energy for movement, for its journey. And then there's the tail. Most of the tail is a flagellum, group of contractile filaments that produce whip-like movements to propel the sperm forward. Now, during ejaculation, hundreds of millions of sperm are released into the vagina, but millions are blocked from entering the uterus by the cervical mucus. We covered this in the uterine cycle, how the consistency of the cervical mucus changes as a result of estrogen and progesterone concentration. When there's high levels of progesterone, the cervical mucus becomes thick and sticky, creating that mucus plug, and that's to prevent any bacteria from entering, just in case fertilization has occurred, to protect the growing embryo. And other reasons why millions of sperm don't make it is due to the acidity of the vagina. The vagina is acidic to protect the environment, and the journey is quite long, and so it needs a lot of energy, so millions of sperm don't make it. And so when sperm is released, it can enter the uterus within minutes of ejaculation. But sperm can survive within the cervical mucus for a day or two, and then they can be transported to the uterus. So only about 100 to 200 sperm actually make it to the fallopian tubes. So how is sperm transported then? Sperm is transported into the fallopian tubes by uterine contractions and also via the sperm's own propulsions. They're like mermaids. They've got tails for a reason, to produce whip-like movements and travel to the fallopian tubes. The sperm are not able to fertilize the egg until they are acted upon by secretions of the female reproductive tract. Fluids in the tract prepare the sperm for fertilization. So now let's subtract complexity and break down fertilization. Fertilization has 4 major steps, and the first step is the process called capacitation or priming. This process enables the sperm to have the capacity to fertilize the egg, because if they reach the egg before capacitation is finished, they will be unable to penetrate the egg. Capacitation causes several changes. Number 1, increased sperm motility. So the tail movement changes from a regular bead-like action to a whip-like action to help propel the sperm forward. They're like mermaids. The second is the sperm's plasma membrane changes. The protein coat of sperm is removed, so it can release lysosomal enzymes, digestive enzymes capable of destroying the egg's outer layers, the protective layers, so the sperm can fuse with the surface membrane of the egg. So that's the first step, preparing the sperm for penetration. The second step is binding, or penetration of the protective layers. Recall that the secondary oocyte is surrounded by 2 protective barriers or layers. The outer layer is the corona radiata, and then there's the zona pellucida. The sperm cells need to penetrate the corona radiata, the first barrier, and sperm cells are able to do this through the release of digestive enzymes that then digest the extracellular matrix of the corona radiata, and then they can use their flagellum to go through. Now, the first sperm to reach the egg is never the one to actually fertilize it, because hundreds of sperm help break down the corona radiata. I only have 1 sperm here, but think that there are hundreds attached, okay, helping break down the corona radiata, these granular cells, and of course the zona pellucida as well, to create a path so that 1 sperm can bind with the plasma membrane. It's teamwork. Well done, boys. And once the granular cells are degraded, sperm cells reach the second barrier, the zona pellucida. And in order for the sperm to penetrate the zona pellucida, the head of the sperm binds to sperm receptors on the zona pellucida, receptor ZP3. Recall that the zona pellucida glycoproteins are receptors for sperm surface proteins. So these here are surface proteins, and this binding triggers the acrosome reaction. The acrosome reaction involves the release of hydrolytic enzymes from the acrosome, and these enzymes are going to break down the zona pellucida. It's going to create a pathway for the sperm cell to reach the plasma membrane of the egg. Let's go through this again, okay? So the acrosome, this here, is the protein-filled vesicle located at the tip of the sperm, and the acrosome is going to burst, okay? And the sperm's plasma membrane fuses with the membrane surrounding the acrosomal vesicle, and it's going to create an opening. And so now the contents of the acrosome, the enzymes, are going to be exposed to the zona pellucida. Then the digestive enzymes of the acrosomes are released. The content of the acrosome are released. And the components such as acrosin and the serine protease, these digestive enzymes drill a passage. They drill this pathway here so that the sperm can reach the egg. It uses its tail to go through the egg's coating. They're like mermaids, using their tails to propel themselves forward. The first sperm to penetrate the zona pellucida and reach the plasma membrane of the egg fuses with this membrane. So as we can see here, it's going to fuse. Their membranes are going to fuse. And the head and midpiece of the sperm enter the cytoplasm. And then we reach step 3 of fertilization, fusion of the sperm and egg. Once the sperm fuses with the egg, the tail stops moving, and the sperm enters the egg by elongation and fusion of the egg's microvilli. And the sperm's nucleus enters the egg's cytoplasm. Now when the first sperm fuses with the oocyte, the oocyte immediately activates mechanisms to prevent polyspermy. So polyspermy is when the egg is fertilized by more than one sperm. And this produces zygotes with more than a diploid amount of DNA, leading to embryonic defects, and it's incompatible with life. And there are 2 types of mechanism. The first is what's known as fast block. This is where there's an immediate change in sodium-ion permeability, and this depolarizes the plasma membrane of the egg, preventing the binding of any other sperm cells. It lasts for about 1 minute, and during this time, there's going to be an influx of calcium ions that occurs following penetration of the sperm, and it's going to stimulate the second mechanism that prevents polyspermy, and this is what's known as slow block. The slow block is also referred to as the cortical reaction, okay? So what happens is there are these cortical granules that sit below the plasma membrane of the egg, and immediately it's going to fuse with the membrane, and it's going to release zonal inhibiting proteins and mucopolysaccharides into the space between the oocyte plasma membrane and the zona pellucida. So these little guys here, these inhibiting proteins, they cause the release of any other sperm that are attached to the egg and destroy the egg's sperm receptors. And so that way, no other sperm can bind. So these zonal inhibiting proteins that are released by the cortical granules are going to release any other sperm that are attached to the egg. And it's also going to destroy sperm receptors so that no other sperm can bind to the egg. So let's go back to the sperm here that has penetrated the egg. Once it's in the cytoplasm, the sperm's nucleus forms a new nuclear envelope called a male pronucleus. And the male pronucleus will use the microtubules to move to the center of the cell, where it fuses with the female pronucleus and form a diploid nucleus. And this leads us to step 4 of fertilization, the formation of the zygote. So I mentioned earlier, and we covered this in the oogenesis lecture, that the oocyte has not yet completed meiosis. All secondary oocytes are frozen in metaphase 2. They are rested in metaphase 2 until fertilization occurs. Now that the egg is fertilized, it completes meiosis 2 and becomes a mature ovum, a female haploid gamete. And along with the mature ovum, a second polar body is formed, but it's non-functional, so it will disintegrate along with the first polar body that was also produced. So now the 2 haploid nuclei derived from the maternal side and paternal side, the sperm and oocyte that are contained within the cytoplasm of the egg are referred to as pronuclei. They're going to decondense their genetic information and replicate their DNA in preparation for mitosis. The pronuclei will then move towards each other, and their nuclear envelope will disintegrate, and their genetic material will combine to produce a single-cell diploid zygote, which contains 46 chromosomes, 23 from the egg, the maternal side, and 23 from the sperm, spermatozoa. So now it has all the genetic information to initiate embryogenesis, and this completes the process of fertilization. Okay, let's go back to this diagram here. The egg is ovulated and sperm travels to the fallopian tubes to fertilize the ovulated oocyte and forms a zygote. Now, the zygote will remain in the fallopian tube for 3 to 4 days, and that's because of the smooth muscle contraction that's occurring in the uterus, induced by estrogen. And so the smooth muscle needs to relax first to allow the zygote to move into the uterus. Let's go through when and how this happens. So the smooth muscle relaxes when the concentration of plasma progesterone increases, and this increases when the corpus luteum develops and starts producing large amounts of progesterone to prepare the endometrium for a possible pregnancy. So while the zygote is in the fallopian tube, it will undergo several mitotic cell divisions. This is a process known as cleavage, although there's no cell growth. So once the zygote, a 16 to 32 cell, reaches the uterus, it's still the same size as it was originally when it was fertilized, and each of these cells is totipotent. They are stem cells. Okay, now usually a female releases only one egg during an ovulation cycle. Only one egg is selected to become the dominant follicle that is ovulated. However, it is possible that 2 eggs are released and fertilized by 2 different sperm. So 2 zygotes form and develop, leading to the birth of dizygotic twins or fraternal twins. Okay, and there are some occurrences where a zygote can split or divide into 2 separate offspring. This usually happens during the early blastocyst stage, resulting in the birth of monozygotic twins or identical twins. And that's how twins are created. Either 2 eggs are ovulated and fertilized by 2 different sperm. This leads to dizygotic twins or fraternal twins, or the zygote splits, usually in the early blastocyst stage, leading to monozygotic twins or identical twins. And once a developing human reaches the uterus, it will continue to undergo several divisions to approximately 100 cells, and the intrauterine fluid provides the growing embryo with nutrients, and then it reaches the stage known as blastocyst, and the cells will start differentiating and implantation will soon occur, which is a topic for another lecture, and that is fertilization. But before we end this lecture, let's quickly summarize everything we've covered. Okay, so the secondary oocyte is arrested in metaphase 2 of meiosis 2, and it's ovulated around day 14 of the menstrual cycle, and ovulation is triggered by a mid-cycle surge of luteinizing hormone known as the LH surge, and fimbriae, which are the finger-like projections at the end of the fallopian tube, will sweep the egg towards the tube. The tube is lined with ciliated epithelium, so it helps the egg move towards the uterus. It takes about 4 days for the egg to reach the uterus. And the secondary oocyte will complete meiosis 2 once it's fertilized. But because of its short viability of 24 to 48 hours, it's got a best before date, release of sperm into the reproductive tract needs to occur between 5 days before or 1 to 2 days after ovulation. Now, hundreds of millions of sperm are released during ejaculation, and within minutes it reaches the uterus. However, millions of these sperm don't make it. Only about 100 to 200 sperm make it to the fallopian tubes. There are several reasons for this: the acidity of the vagina, blocked by cervical mucus, and the journey is just very long and it requires a lot of energy. That's why a lot of sperm don't make it. And fluids in the female reproductive tract prepare the sperm for fertilization, a process known as capacitation or priming, which increases sperm motility and changes the sperm's plasma membrane so that it's able to penetrate the egg and fuse with the surface membrane. Acrosomal reaction also occur where the acrosomal vesicle fuses with the sperm's plasma membrane so that the digestive enzymes are exposed to the zona pellucida. Many sperm bind to sperm receptors on the zona pellucida, and so the zona triggers the acrosome reaction and the sperm is able to move through the zona pellucida using their tail to propel themselves forward. So that's the first stage. The second stage is the sperm binds to the egg, and the egg activates mechanisms to prevent polyspermy, such as the cortical reaction, where the cortical granules inside the egg fuses with the membrane and release zonal inhibiting proteins that release any other sperm that are attached to the egg and also destroys sperm receptors so that no other sperm can bind to the egg. Then the sperm is drawn into the egg its nucleus enters the cytoplasm and the oocyte completes the second meiotic division and develops into a mature ovum. And along with the mature ovum, a second polar body is produced, but it's small and non-functional, so it will eventually disintegrate along with the first polar body. And then the nuclei of the sperm and egg combines, creating a pronucleus, and a zygote is formed and embryonic development will begin. So that's fertilization. In this lecture, we learned the structures of the female and male gamete and how they are transported. We broke down how the sperm needs to be prepared, a process called capacitation, before it's able to penetrate the egg. We talked about how the sperm penetrates the two protective layers, the corona radiata and the zona pellucida of the egg, and how it fuses with the egg's membrane. We also covered the two mechanisms that prevent polyspermy, fast block and slow block. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating.